This is the beginning of Unit 1, Part 4. Now, some molecules are polar and some are nonpolar. A nonpolar molecule, when you look at the sharing of electrons, it's equal between the two or more atoms, all the atoms within it. If there's unequal sharing and the electrons spend more time around one atom than the next one over, around one atom nucleus as opposed to the other atom that they share with, they're being shared with, then it's a polar molecule. So nonpolar is equal sharing and polar means unequal sharing. The unequal sharing means that the one nucleus of the atom that they spend more time around is slightly more negative, and that gives it its polarity. Typically, if you have a larger number of valence shells, they're more electronegative, and if the electrons are in atoms with fewer valence shells, they're more electropositive. I'm not sure if I can show a good example of that. It's the unequal sharing that produces polar molecules. They're polar, they have a unequal distribution of charge around them, and you'll see what that means. Here, and it's extremely important in biology, the polarity of water makes all kinds of interactions possible. In fact, the polarity allows for hydrogen bonding, and you'll see how important that is in proteins and in DNA molecules. Okay, so carbon dioxide is a nonpolar molecule. The electrons between just a second. The electrons of this oxygen that are shared in the double bond with this carbon are shared equally. There is no slanting more to one atom or the other. Same goes for the electrons shared with this oxygen forming that double bond. The electrons are equally shared between that oxygen and that carbon, or that carbon and that oxygen. And so there is no polarity. However, in a water molecule, in water, there are many other examples of polar molecules, but water is a really good one to talk about. In a water molecule, with this oxygen, the electrons in that bond there, and also in this bond here, that are shared with this hydrogen, between this hydrogen and the oxygen, and between this hydrogen and the oxygen, the electrons spend a little bit more time with the oxygen as opposed to these hydrogens, making this molecule polar in that it has polarity, it has a pole, it has a more negative area around the oxygen, and around the hydrogens it's more positive. Now you're going to see in a minute what comes out of that, what happens because of that. And this is just a summary slide looking at ionic bonds versus polar covalent versus non-polar covalent. Remember, in covalent bonding you have a sharing of electrons. In an ionic bonding you have a transfer of electrons. And here you see the non-polar example of carbon dioxide, and here you see a polar example of water molecule, where one end of the molecule is more negative. So it has polarity, it is polar. Well, we talked about hydrogen bonds a little bit. We talked about three types of bonds: ionic bonds, covalent bonds, and hydrogen bonds. We didn't really get into much detail about hydrogen bonds. They're, as I did say earlier, they're quite weak. They do bind, they can bind, help bind together or hold together atoms, but they are extremely weak and easily broken apart. It's typically the only time that hydrogen bonding will cause great stability in a molecule is if there's many, many, many hydrogen bonds between different parts of the molecule or between different strands. like a DNA, for example, has two strands held together by hydrogen bonds. These are typically what you find with water solutions, that there's a dipole in the molecule in H2O, and it causes actually an attraction between the water molecules, the H2 molecules in the water. And that is what is responsible for surface tension. Water has a surface tension. Insects can walk on it. The molecules at the very surface are held more tightly together. It allows for intramolecular bonding to form in large three-dimensional molecules, like the DNA molecule, as you know, is a double-stranded molecule. It's a very long double strand, two strands. They are not covalently bound with cross-bridging. Yes, there is cross-bridging. You probably learned that, but each cross-bridge is held together right in the middle there by not covalent bond, but by hydrogen bonding. But there are so many that the two strands become very stable with each other. Here you see an example of why why the polarity, or it's called a dipole. This is the letter delta, the Greek letter delta, written like that. And when it has a negative sign, it shows a negative polarity. The electrons are spending more time at this end, and the hydrogen atoms in the H2O molecule have a positive delta, or positive polarity, and negative and positive do attract each other, but this is already in one molecule. But because negative and positive attract each other, this negative charge here will be attracted to the partial positive dipole, the dipole charge on this hydrogen, and therefore you get this, and it's shown as dots, hydrogen bonding, because the whole thing is surrounded all over the place with many, many water molecules. They arrange in such a way that they will form many hydrogen bonds as interactions between these hydrogen and oxygen, because of the dipole on each one, positive and negative dipole. Now, chemical reactions will occur between atoms or between molecules if bonds are formed, rearranged, or broken. And here we're talking about ionic bonds and covalent bonds, mostly covalent bonds in biology, but sometimes ionic bonds. And a chemical reaction is written in a particular form. It's written as an equation, and the equation contains the number and type of reacting substances, either molecules or atoms, elements on one side and it'll show the relative amounts of the reactants and the product. So you have like molecule A plus molecule B going towards forming something called A. There's the covalent bond, and B, right? More commonly just shown as AB with the covalent bond inferred. Many reactions in biology are in fact reversible. Not all, but many are reversible, and that's shown by this double-headed arrow. Here you see a reaction between two hydrogens to form hydrogen gas. Here you see 4 hydrogens and carbon forming methane. So the equation is this way in terms of the product is on this side at the end of the arrow. These are products. There are different patterns. There are combination reactions where two things come together to form one. You have decomposition or breakdown, where something gets broken down into its individual smaller molecules. Sometimes you get an exchange, A, B plus C. In this particular reaction becomes AC plus B. Okay. You should understand that in biology there are much more complex, larger molecules. There are actually 4 major kind of molecules, which we'll talk about soon enough. In biological systems, all living biological things have 4 main categories. One of them, one of the four categories are protein molecules. And protein molecules are formed by, it's a polymer. Polymer is a strand held together of many individual building blocks. And the building blocks for proteins are amino acids. So each of these spheres represents an amino acid. And when it's formed into a protein, it can form this long chain. This is just a simple example of a protein made up of 2, 4, 6, 8, 10, 12 amino acids that together form a polymer to form a polymer chain protein. 12 amino acids going to form a polymer. Here you see glycogen, and I already mentioned glycogen when I talked about insulin in Unit 1. Glycogen is a polymer of glucose molecules. They are held together just like the protein is held, our amino acids held together by covalent bonds. In glycogen, you have covalent bonds holding together glucose. Remember, I told you that glycogen actually is found, one of the places is found is in the liver, and it acts as a storage form. So the liver is a storage site for glycogen, and when blood glucose levels drop, then these bonds here and here, etc., at the ends of these chains, so gradually working inwards, they'll get broken, and the individual glucose units or glucose molecules get released in the bloodstream, thus raising the glucose level in the bloodstream. But the main idea here is you see the building up of a polymer from individual molecules, the formation of covalent bonds to form this polymer structure, and here they're being broken apart. The polymer glycogen is being broken apart into individual glucose molecules. Here you see another type of reaction, an example of an exchange reaction. In this case, you have a glucose molecule, and it's reacting with adenosine triphosphate, ATP, which, as I mentioned earlier, is the storage form of the chemical molecule that acts as a chemical storage form for energy in living tissues and cells, because these covalent bonds between the phosphates contain tremendous amounts of energy. What happens in an exchange reaction in this case, and these are the two reactants, glucose in this case, in this reaction, glucose and ATP are interacting together to form the product glucose phosphate. So you have an exchange of one of the phosphates, this terminal phosphate, it's transferred over to the glucose to form a molecule called glucose phosphate and adenosine diphosphate. That's kind of a nice naming. Sometimes they're named clearly. So here, referring to the 3 phosphates, triphosphate, and here di referring to the fact that it has 2. Once you take a molecule, let's say, for example, any molecule, but in this case, in this example, this molecule of ATP, and you either add something or remove something, either forming a new covalent bond is formed here. In this one, you're forming a new covalent bond there. Or if you remove something, you break a covalent bond and the molecule is changed, then you have a new molecule with a new, different name and new physical and chemical properties. All right. Now there are certain classes of reactions that are quite important in biology, particularly during energy formation. And as you know, chemical energy is stored in cells as ATP. So this class of reactions called oxidation reduction reactions. In short form, they're called redox reactions. They are important in a lot of processes, particularly in the generation of ATP within cells. During an oxidation reaction, the reactants that lose electrons, they're giving electrons away, so they're considered electron donors, and they're becoming oxidized. So oxidation is a loss, all right? Oil. Oxidation is a loss of electrons. In an oxidation reaction, they lose electrons, they donate electrons to something else. If you're looking at an oxidation reduction reaction or redox reaction, and the reactants take up in the reaction, the molecules are taking up electrons, or they're acting as electron acceptors, then they're becoming reduced. Okay, and reduction is a gain of electrons. So these are called, for short, and it's a way of remembering, oxidation is a loss, and reduction is a gain of electron. Oxidation is a loss, reduction is a gain. Oil rig. So very typically you have, in these kind of reactions, oxidation reduction reactions, you have two things, one of which loses electrons and the other one gains electrons. They're usually paired reactions where you're getting loss, gain, and it can be a whole series of things. One molecule gives up an electron to another molecule, so that molecule now has more electrons, and it donates electrons to the next one in a chain. You know, it's a chain of reaction, a chain of reactions. Okay, in general, when you look at reactions that release energy, we'll get back to oxidation reduction reactions later on in the course. In general, when you look at reactions where you have energy flow, energy changes, if you get a release of energy during the reaction, then it's called an exergonic reaction. If the molecules have to take up energy and therefore they contain more, the final products contain more energy than the initial reactants, the molecules that were reacting to carry out that reaction, to form that end product, then it's called, if they're taking up energy, then it's called an endergonic reaction. As I said earlier, many reactions often are reversible. So a reaction like this could go in the opposite direction and could be written like this: A plus B. And what I'm drawing here is the exact equivalent to what's written on the left-hand side. Just put in a double-headed arrow, and you get A plus B can go to form AB, but AB can also reverse and split the covalent bond that's between the AB and form A plus B. And these are in a constant state of flux, shifting one way, back and forth. If neither the forward nor the reverse reaction is dominating, then the equation is said to be in chemical equilibrium and is at a steady state. A plus B does go to AB to form AB, but AB is breaking down at at an equivalent rate to form A plus B. Often in chemical reactions, what you find is if the constituents, the parts of the molecules that are involved on one side of a reaction are in higher concentrations, so if A plus B are in higher concentrations, then the reaction will tend to go this way and going towards forming AB. It'll be mostly going towards forming AB. And conversely, if there's more AB than A plus B, then the reaction will tend to go in the opposite direction for many chemical reactions, and we'll talk about that later on in the course, examples of that happening. All right. The factors that influence the rate, and I just mentioned concentration. That's one factor, but these are a whole series of factors that are all important, all contributing to the rate or speed of chemical reactions. Temperature is important. Chemical reactions typically proceed faster when energy is put in at a higher temperature. Particle size is important. Smaller particles can move around faster and interact more quickly to speed up, and therefore have a faster reaction rate. Concentration, as I said, the higher concentration will produce faster reactions. It could be on the left side, the right side, and you get these shifts one way or the other. Catalysts are very important. These tremendously increase the rate of reaction without themselves being changed. By the end of the reaction, you still have the catalyst there. It doesn't go into forming the final product, but speeds up the reaction. And now in biological systems, the catalysts are called enzymes. Virtually all catalysts in biological systems, not 100%, but close to 100% of them are proteins. They're made up of amino acids. Okay, and different chemical reactions, biochemical reactions in biological systems typically do require enzymes in order to act at a speed fast enough for biological systems to work, for a cell to actually have a normal production of the things that it needs. So enzymes are absolutely required, and there are many, many different enzymes in biological systems because for each reaction, there's a specific enzyme. If two things are going to react together in biological systems, typically you do have it, not always, but typically you do need an enzyme catalyst. So enzymes are biological catalysts. They are proteins. They speed up reactions, and they are very specific for a particular reaction. Okay. In biological systems, we have biochemical reactions. We have mostly organic compounds being formed or broken down, but we also have some more inorganic compounds. All organic compounds contain carbon within them. All are quite large usually, and some are tremendously huge molecules. The carbon is covalently bound into the molecule. It forms part of the molecule. So things like carbohydrates or proteins, lipids, DNA, and nucleic acids, all of those, which are actually the four major types of molecules found in living things, are all organic compounds. These are molecules that are involved in biological chemical reactions. Biochemistry, in other words. There are some also inorganic compounds that are involved. They're very important. Inorganic compounds do not contain carbon. Some examples of that are water or salts. And salt does not mean necessarily just NaCl. It's any two ions that form ionic bonding with each other. Calcium chloride. There's an example of a salt. But the major ionic salt in living systems is NaCl. But salt, we use salt in day-to-day to refer to NaCl, but it is not the only type of salt out there. Also, many of the acids and bases that are found in living systems are inorganic. They don't contain carbon. Inorganic compounds include water, and 60 to 80% of cells are made up of water, and salts, as I said, and acids and bases. We'll go through each of these. There are certain key characteristics, chemical and physical characteristics of water that lend themselves to living systems that allow us to be alive. Water is a key. It is our solvent. It has these properties include the fact that it has a high heat capacity. It will absorb or release large amounts of heat before it will be able to change its temperature. It has a very high heat of vaporization. In order to change phases going from liquid to gas, it has to take in or acquire or absorb a lot of heat. In order to change from gas to liquid, it has to release a lot of heat. The same thing goes, by the way, when water goes from liquid to solid frozen ice, although that doesn't happen normally in a living biological system, not humans. Water is a polar liquid, and polar solutions are good solvents, and that means that they can easily dissolve molecules. And water is one of the important aspects of water is a lot of things can, a lot of molecules can dissolve into it. And the reason is because it's polar. So polar solvents will easily dissolve ionic substances like calcium chloride or NaCl. It can act as an excellent solvent. It will form hydration layers around large charged molecules. Hydration layer means that you have the molecule there. Okay, let's say that's an atom. There's a covalent bond there. There's another atom making up the molecule. There's another atom. There's two covalent bonds sticking that way, you know. And if this is a polar molecule, then that means it has a charge. If it has any charge, then it's polar. Let's guess that this one has a negative charge, more negative at that end than at this end, slightly more negative at that end than this end. Maybe there's another positive charge here. All of these can interact with the water molecule that is itself polar. Remember, it has a dipole. So water, if you look at water, if that's the oxygen, and these are two hydrogens, then remember that the oxygen has a negative dipole, slightly negative, and the oxygen and the hydrogens are slightly positive. So they'll arrange themselves, you know, that in such a way, this water molecule would arrange itself in such a way that there would be a hydrogen near that negative end, and oxygen would be turned towards that positive because positive and negative interact. So it acts very well to form a hydration layer and to dissolve things because they'll break apart, they'll go into solution, they'll be completely surrounded by water. Each individual molecule of this substance will be completely surrounded by water because of the polar solvent properties of water. Water also an important property is the fact that it is reactive. It's involved in what are called hydrolysis and dehydration reactions, and you'll see examples of that later. Another physical property is it can act in cushioning around certain body organs. Okay, salts like NaCl will contain cations other than hydrogen and anions other than hydroxyl group. Hydrogen ion and hydroxyl group together, these form water, of course. If it's anything else made up of cation and anion held together by an ionic bond, then it's a salt. And solutions with these kind of salts act as the salt acts as an electrolyte. It conducts electrical current. Acids and bases. The characteristic about acids and bases are very important in chemical reactions because you need a certain level of hydrogen ions in order for reactions to occur. For different reactions, you need a certain hydrogen ion level or a hydroxyl group level. Acids can release hydrogen ions. Bases can release hydroxyl group, OH-. So here you see an example here of, this is HCl. HCl is hydrochloric acid. It has the ability, it's very reactive and has the ability to release hydrogen ions. It's an acid. It is acting as proton donor. That's what acids do. This makes it highly reactive. The level of hydrogen ions in a solution is what determines, in fact, the pH of that solution. Okay? Bases will release a hydroxyl group. Sodium hydroxide is a strong base because it can release hydrogen hydroxyl anion. Hydroxyl anion. Yeah, no, hydroxyl group. All right, we'll get into acid and bases probably a little bit more, certainly later. Perhaps later is now, we'll see. So, acidic solutions have a higher concentration of hydrogen ions, and when you look at the pH, they have a lower pH. So the lower end of the pH scale. Remember, the pH scale goes up to, I think it's 14, from a pH of just about 0, above 0 to 14, right? And the higher the number, the more basic, the lower the number, the lower the pH number, the more acids. So acids have a lower pH value, and base solutions, basic solutions, or alkaline, basic or alkaline are synonymous. They mean the same thing. You can use the word basic instead of alkaline. So basic solution is an alkaline solution. BAS, it doesn't come out very well here, does it? Let me get the black pen. Basic solution. Basic is the same as alkaline. A basic solution will have a lower hydrogen ion concentration. Where is it? A lower hydrogen ion concentration, and therefore a higher pH. Yes, I did say that alkaline solutions, a basic solution or alkaline solution, or compound releases hydroxyl anions, but when it releases the hydroxyl group, OH minus, that will interact with any hydrogen ions present, and therefore there'll be fewer. So a basic solution will have a lower hydrogen ion concentration, and when you look at the pH scale, that translates into a higher pH. And here you see the acid-base pH scale showing the concentration, and the important key thing to realize here is that this scale is a logarithmic scale. Essentially neutral, neither basic nor acidic solution would have would be around a pH of 7. If the solution has a pH of 6, a lower pH, then it contains more hydrogen ions. And the difference between 7 and 6 is a 10-fold. Each step here between 7 and 6, here and here, 7 and 6, each step is 10-fold. So you have a 10-fold increase in hydrogen ion concentrations. Between 7 and 8, you have a 10-fold decrease level of hydrogen ion concentrations. These are tremendously basic. They will, well, at least down here certainly, they can damage tissue. Up here, they're very acid. They can damage tissue. In most biological systems, you're going to have pH of about 7.4 with some range around that. There are exceptions to that. In the space of the stomach, you have a lot of hydrochloric acid. The pH can be as low as 2 or even lower. And if you think about it a bit, that's should not be difficult for you to understand that helps break down food, helps to kill off bacteria that might be in the food, and protect the individual from infections in terms of the stomach acidity. Buffers. And again, and this is the last slide, all of this stuff you should have touched on in quite a bit of detail before here. I would expect this to not be the very first time that you've heard about these basic fundamental chemical principles, and you have already some reasonably good exposure to this. Buffers act to prevent large changes in pH, and buffers are extremely important in different body fluids. main buffering system, not the only buffering system, and you will learn a lot about buffering systems next semester when you do renal physiology, but we will do this somewhat this semester when we do respiration and blood. But the major buffering system in the body is called the carbonic acid bicarbonate buffering system. This is bicarbonate, okay, HCO3 minus is bicarbonate. This is carbonic acid, and you get a shift between carbonic acid and bicarb and hydrogen ions. Carbonic acid can dissociate. It's reversible. You see there's a double-headed arrow there, so it can go back and forth, and will release bicarbonate ions and proteins. The chemical equilibrium between them helps to resist pH changes, because if you have more hydrogen ions going in, which could make the solution more acidic, it will tend to bind to the bicarb and form carbonic acid, which is quite a weak acid. And that's the end of Unit 1, Part 4.